Welcome, everyone. Today, we are diving into a highly precise molecular technique known as the allele-specific oligonucleotide dot blot. If you have ever wondered how doctors can look at a patient's DNA and pinpoint a single mutated letter that causes a genetic disease, this is exactly how they do it. It is a brilliant, elegant, and surprisingly simple tool that forms the cornerstone of modern genetic testing. Let us begin our journey into understanding how this molecular magnifying glass actually works. Before we get into the heavy scientific terminology, let us build an intuitive understanding of what we are trying to achieve. Imagine you are tasked with finding a single misspelled word in a massive encyclopedia containing billions of letters. Reading every single page, line by line, would take a lifetime. In the world of genetics, our DNA is that massive encyclopedia. Sometimes a single wrong letter can cause a serious genetic disease. The allele-specific oligonucleotide dot blot is exactly like a magical glowing bookmark. Instead of reading the whole book, you drop this bookmark in, and it only sticks to that exact misspelled word. It allows scientists to find a single mutated DNA letter among billions of normal ones, quickly and accurately. Now that we understand the goal, let us break down the formal scientific name of this technique so it makes perfect sense to you. First, an allele is simply a specific version of a gene, such as a normal, healthy version or a disease-causing mutated version. Next, the word oligonucleotide refers to a very short, artificially created piece of single-stranded DNA, usually about 15 to 20 letters long. Finally, a dot blot describes the physical laboratory method itself. We literally place a tiny drop, or dot, of a patient's DNA onto a special nylon membrane. So, putting it all together, we are using a short, custom-made DNA probe to stick to a specific genetic mutation spotted on a membrane. Building on those definitions, let us look at how this mechanism actually works in the laboratory. Once we have spotted the patient's DNA onto the nylon membrane, we introduce our custom-made oligonucleotide probe. This probe is designed to be the exact complementary match to the mutation we are looking for. If the probe matches the patient's DNA perfectly, it sticks tightly and creates a visible dark spot on the membrane. However, the magic of this technique lies in its strictness. If there is even a single letter mismatch between the probe and the patient's DNA, the binding is too weak. During the washing step, the mismatched probe washes away completely, leaving a blank space. This all-or-nothing binding is what makes the test so incredibly precise. Understanding how strict this binding is, you might be wondering why we need such a specific tool when we have modern DNA sequencing machines available today. The reality is that sequencing an entire gene is often expensive, time-consuming, and completely unnecessary when we already know exactly which mutation we are looking for. This technique is incredibly important for diagnosing well-known genetic disorders like sickle cell anemia and cystic fibrosis, which are caused by a single incorrect DNA letter. Without this method, rapid and cost-effective screening of newborns or expecting parents for these devastating diseases would be much more difficult. It empowers doctors to give families life-saving answers in a matter of hours rather than weeks. To achieve that perfect all-or-nothing binding we just discussed, we have to look at the thermodynamics of DNA. DNA strands stick together because of hydrogen bonds, and the temperature at which half of these bonds break is called the melting temperature. We estimate this critical temperature using a simple mathematical formula known as the Wallace rule. In this formula, Tm represents the melting temperature in degrees Celsius. The variables A, T, G, and C represent the total number of adenine, thymine, guanine, and cytosine bases in our short DNA probe. Guanine and cytosine pairs form stronger bonds, contributing 4 degrees each, while adenine and thymine pairs are weaker, contributing only 2 degrees each. If we increase the number of G and C bases, the melting temperature increases, meaning the DNA strands hold together much tighter. We must wash the membrane just below this temperature.
Now that we have our formula, let us see this with a concrete example. Suppose we are working in a diagnostic laboratory and have designed a 14-letter probe for a normal gene. The sequence is given on screen. We need to calculate its exact melting temperature to determine the correct washing temperature. First, we carefully count the bases. We find exactly 3 adenines, 3 thymines, 4 guanines, and 4 cytosines. Next, we substitute these counted values into the Wallace rule formula. We group the weaker adenine and thymine pairs together, giving us 2 times 6. Then we group the stronger guanine and cytosine pairs together, giving us 4 times 8. Adding 12 and 32 gives us a final calculated melting temperature of 44 degrees Celsius. This means we must wash our dot blot membrane at exactly 44 degrees Celsius, so the perfectly matched probe remains bound while any mismatched probe washes away.
With the physical washing steps calculated and understood, we can now interpret the actual visual results of a dot blot test. When we test a patient, we probe their DNA against both a normal sequence and a mutant sequence. We represent their overall genetic makeup, or genotype, using a simple additive formula. In this formula, G represents the overall genotype of the individual. The variable AN represents the normal, healthy allele, and AM represents the mutant, disease-causing allele. Because humans inherit one copy of a gene from each parent, the overall genotype G will always be the sum of 2 alleles. If a patient's DNA binds to a specific probe, it means they possess that specific allele. By adding the visual evidence from the normal and mutant membranes together, we can perfectly deduce their exact genetic makeup. Let us apply this genotype formula to a real-world diagnostic example. A couple is expecting a child and wants to know if they are carriers of the sickle cell anemia mutation. The doctor spots their DNA on 2 separate membranes, one probed with a normal ASO and the other with a mutant ASO. We observe that the mother's DNA shows a dark spot on both membranes. What does this mean? A visible spot on the normal membrane means she has at least one healthy copy of the gene, which we write as AN. The spot on the mutant membrane means she also possesses at least one mutated copy, AM. Combining these 2 pieces of visual evidence, her genotype equals 1 normal allele plus 1 mutant allele. Physically, this means she is a heterozygous carrier. She is healthy, but carries the hidden mutation. Now that we have deduced the mother's genetic makeup, let us analyze the father's results. His DNA was subjected to the exact same procedure. However, his visual results are different. His DNA only bound to the normal probe, showing a dark spot on the normal ASO membrane. This gives us our first allele, AN. Crucially, he showed absolutely no signal on the mutant probe membrane. Because humans must have 2 alleles for this gene, and he has no mutant alleles, his genotype must consist of 2 normal alleles. We represent this by adding AN to another AN. Physically, this indicates he has 2 healthy copies of the gene and is homozygous normal. He does not carry the sickle cell mutation at all. So, we have successfully deduced the genotypes of both parents. This brings us to the most important question. Is their unborn baby safe? Let us prove this logically. We know the mother is a carrier with one normal and one mutant allele, and the father is completely normal with two healthy alleles. A child must inherit exactly one allele from each parent. To actually suffer from sickle cell anemia, the baby would need to inherit the mutated gene from both parents, resulting in a genotype of AM plus AM. However, looking at the father's genotype, he only has normal alleles to give. He can never contribute a mutant allele. Therefore, the worst-case scenario is that the child inherits the mother's mutant allele and the father's normal allele, making them a healthy carrier. We can confidently prove and tell the parents that their child will not have the disease. To reinforce everything we have just learned and proven, let us look at this comprehensive diagram. You are seeing the 3 possible genetic states a patient can have and how they visually appear on a dot blot. On the left, we have the membrane probed with the normal ASO, and on the right, the mutant ASO. Look at the top row. A homozygous normal individual, like the father in our example, shows a dark spot only on the normal side. The middle row shows a heterozygous carrier, like the mother, with dark spots on both sides because they possess both alleles. Finally, the bottom row shows a homozygous mutant individual who would unfortunately suffer from the disease, showing a spot only on the mutant side. This simple presence or absence of dots provides profound, life-changing medical information. We have covered a lot of ground today, so let us summarize the key takeaways. The allele-specific oligonucleotide dot blot is essentially a highly precise molecular magnifying glass used to detect single-letter changes in DNA. We learned that its accuracy relies on the strict rules of DNA base pairing and thermodynamics, where even a single mismatch causes the probe to wash away completely. By calculating the exact melting temperature, we ensure perfect binding. Finally, we saw how reading the simple visual spot patterns allows us to deduce a patient's exact genotype, making this a rapid and cost-effective tool for diagnosing devastating genetic illnesses. You now have a complete understanding of both the biological principles and the practical applications of this amazing diagnostic tool. Great job!